Hello everyone, as you already know, my name is Jose Miguel, and here I am presenting again my review with the following title: Susceptibility and Survival in Idiopathic Pulmonary Fibrosis by means of linkage analysis and association studies. The goal of this talk is to present the main findings of the works by Siebold, Pelto, and North groups, where susceptibility and mortality in idiopathic pulmonary fibrosis were assessed. This talk is divided into 4 main sections. In the first place, I will do a brief introduction to the disease. Then I will proceed with a short section on the experimental setups used by the authors, and continue with the main findings of this review, presenting results and some discussion. And finally, we will end with some conclusions. So, what is IPF? IPF stands for Idiopathic Pulmonary Fibrosis, where idiopathic means of unknown origin or etiology. This is a complex disease with a suspected genetic base, with almost no response to pharmacotherapy. It has a low incidence, but a very high mortality, comparable to those malignancies such as cancer. It belongs to a set of more than 100 pulmonary disorders, but is the most severe chronic form of pulmonary fibrosis. And that is why I have selected this complex disease, because it is a real public health problem. Recent research based on the applications of linkage and association analysis on a genome-wide scale have identified and described novel variants associated with an increased susceptibility and mortality in IPF. So here are the 3 papers I have reviewed. The experimental setup sections of my review has been now structured in several subsections that I will present in the following slides. The 3 papers I have reviewed follow different study designs. However, common patterns are shared among them. The designs are a trade-off between costs and expected results. They use well-planned designs based on previous findings, and they use available Omics technologies compatible with low cost to drive further research. The basic idea is to search for a common and rare variant, if possible, using Omics technologies such as genome-wide linkage analysis, which is based on family data, and association studies based on population scale data, to discover associations with susceptibility and mortality in IPF. The outcomes from these 3 papers are usually indirect associations based on the previous scanning of genomes with tagging SNPs. Genome references were used from known projects as the HubMap project and the OneKey Genome project. As we already know, in a GWAS with thousands of possible tests, we keep only statistically significant values. The threshold used to define a genome-wide significance was 5 multiplied by 10 to the minus 8. Linkage disequilibrium was assessed using R-squared measures, and inheritance models were assumed to compare the odds ratios of the heterozygous rare variants and at-risk homozygote genotypes. And finally, gene expression was assessed by Siebold and North groups. They studied the MYC5B expression correlation between IPF cases and controls, and the correlation between MYC5B expression and genotypes. Now I will proceed summarizing the main findings. Siebold et al. Found a high linkage region in the P-terminus of chromosome 11, where health-forming mucin genes are located. They are the genes responsible for mucus formation. And after discovering this region, the region depicted with a blue rectangle in the diagram, they did a fine mapping. The idea behind the LOD score is to study if an allele is transmitted more or less than we would expect by chance. So then we plot the resulting LOD score against the chromosome position and study the presence or absence of the linkage signals. In the diagram we see the linkage signals in chromosome 11, 14, and 17. Linkage disequilibrium analysis based on the R-squared correlation coefficient was used to assess the association among observed variants in this region of high linkage levels. In the diagram, it is shown the linkage disequilibrium analysis based on R-squares of observed SNPs on a pairwise basis. In this diagram, we see the health-forming mucin region at the top, showing several SNPs, and inside the diagrams, the R-square values. The diagram in this slide shows the P-terminus of chromosome 11 in panel A, see the green rectangle, where Siebold identified a region with high linkage level. In B panel, the fine mapping is presented within the health-forming mucin region, and as a result, a variant in the putative promoter of MUC5B gene was identified with a genome-wide significant level. Signals associated with IPF susceptibility in Noth et al. Are shown in this table, and data are presented for the overall 3 stages GWAS, thus overall p-values are presented. MUC5B and TOLIP SNPs showed strongly associated with idiopathic pulmonary fibrosis according to their p-values. IPF individuals who carried minor alleles of the SNPs had effects in the same directions in all GWAS stages, that is, increased risk of IPF or protection against the disorder. Minor alleles of the SNP depicted with a red R constituted a risk for IPF, while minor alleles of SNPs depicted with a green P protected against the disease. Noth et al. Also confirmed the association of the MUC5B variant with susceptibility in IPF found by Siebold in 2011, as well as the novel variants shown on the previous slide. The novel variants at the TOLIP gene loci are not correlated with the MUC5B variant according to the observed R-square low levels. That means that novel variants in TOLIP are independent from that of MUC5B. Regarding the survival in IPF, Pelto et al. Found that the promoted MUC5B variant was related with survival in IPF in both study cohorts, the INSPIRE and the Chicago cohorts. The heterozygous and homozygous for the minor allele T, that is the GT, and the TT genotypes, GT and TT genotypes, were at lower risk of mortality, despite this is the risk allele for IPF. Siebold et al. And Noth et al. Studied the MUC5B expressions in 2 terms. First, the expression and its correlation with disease phenotype, IPF individuals and unaffected individuals, as shown in the slide. And second, the expression and its correlation with MUC5B variant genotypes, which are not shown in the slide. An increase in MUC5B expression was observed between cases and controls, with a factor increase of about 14. So subjects with IPF showed higher MUC5B expression levels than unaffected subjects. When the correlation between the MUC5B expression levels and genotypes was studied, 2 distinct results were observed. Unaffected individuals carrying the GT genotype showed increased expression levels in comparison with unaffected individuals with TT genotypes. And this difference in expression levels with genotypes was not observed in IPF cases. According to Pelto et al., these results suggest the existence at least of 2 different clinical phenotypes, separable by the MUC5B genotype, which are results consistent with a prior report of an association between the MUC5B variant and less severe pathological changes in familial interstitial pneumonia, a phenotype that may be sometimes difficult to distinguish from IPF. Noth et al. Identified, as well as the MUC5B variant in chromosome 11, novel variants in TOLIP in chromosome 14 and SPPL2C in chromosome 17. In addition, the observed effect direction of increased susceptibility and increased mortality risks in IPF for variants in MUC5B and TOLIP genes suggests that these 2 genes are of importance to IPF because they may modify the susceptibility and the course of IPF. And just to conclude, susceptibility and mortality analysis were performed by genome-wide linkage and association omic technologies after considering the trade-off between cost and expected significance of results. A common variant polymorphism located at the putative promoter region of MUC5B gene was found to be associated with the development of IPF and it's also significantly associated with survival in IPF. The finding of more novel variants related to susceptibility and mortality in IPF suggests that more than one gene or loci is involved in this pathology. This result is strongly supported by the independence of variant signals found by North Group. And that's all. I hope you have enjoyed, and thank you for your attention.